Hello everyone! Today we will review the research article Microbiota Changes in Lactation in the Short-beaked Echidna Tachyglossus aculeatus by Isabella Wilson and her colleagues, published by Oxford Academic. Studying microbial ecology of Australian mammals at the University of Adelaide, Wilson is a dedicated microbiologist and PhD candidate. Common themes of her research focuses on how the factors of captivity and reproduction cause shifts in the microbiota that aid in the health of animals. Many of her research findings encourage efforts for conservation, particularly for lactating and non-lactating mammals, along with animals that are wild or held in captivity. Within Wilson's research, microbial diversity was analyzed using concepts we learned in class, such as species richness, which is the number of different species in a community, species evenness, which reflects the relative abundance of each species and how evenly they are represented across a community, the Shannon Index, a mathematical expression that combines both richness and evenness to describe the diversity of a community, taxonomy, which allows for the organization of organisms into evolutionary lineages, such as a phylum, family, genus, etc., and ecological niches, which show the range of resources a type of species can use along with their survival in certain environment conditions. These key ideas allow us to analyze the study's main point of investigation, how the pouch microbiome of echidnas functions as an ecological niche by changing during lactation.
Wilson's study began by examining microbial alpha and beta diversity, shown in this figure. Alpha diversity measures how many different microbes are in one sample, while beta diversity measures how different those samples are from each other. Using metrics like phased phylogenetic diversity, species richness, and evenness, the researchers compared wild and captive echidnas outside of mating and lactation seasons. They found no significant difference between these two. Phase diversity had a p-value greater than 0.05. The unweighted unifrac equaled 0.199, and the weighted unifrac with abundance had a p-value of 0.808. Overall, this means that the microbial balance in wild and captive echidnas was nearly identical. This connects to the concept of community stability, showing that a biological community can remain diverse and functional even when conditions change. The pouch microbiota likely represents a stable, co-evolved group of microbes adapted to both environments.
The researchers also provide an overview of the bacterial species found in the milk of both lactating and non-lactating echidnas, with Figure 4a showing the composition of major phylum and genus classes in wild and captive samples. Across all echidnas, actinobacteria, firmicutes, and proteobacteria dominated. During lactation, firmicutes increased by about 55%, while echinobacteria decreased by roughly 34%. Still, the Shannon Diversity Index showed minimal overall difference, meaning changes were due to shifts in composition rather than total diversity. Figure 4b assigns statistical significance to these findings, showing a Q-value of 0.017 between lactating and non-lactating microbiomes, indicating a lineage-level shift. Since 98% of bacteria came from the three main phyla, the p-value of 0.065 confirms no significant change in overall diversity. This connects to the biological principles by illustrating how lactation shapes microbial niches that protect offspring, linking taxonomy, function, and evolution.
Figure 5 addresses where these microbes come from using Source Tracker 2, which estimates how much of a pouch microbiome originates from sources like the mouth, cloaca, or environment. Since most microbes came from unknown sources, this implies that the pseudopouch develops its own microbial niche. This means that the pouch is not just seeded by external bacteria, but it's a distinct microecosystem shaped by the physiology of lactation and possibly evolved to benefit the young. This niche formation parallels ecological concepts we've studied, how specific environments select for certain taxa and how microbes occupy roles that support host survival. It also shows scientific uncertainty, meaning that future research is needed to identify these unclassified lineages.
Isabella Wilson's research showcases that captivity doesn't have much of a decreasing effect on the echidna's microbial diversity. However, lactation does. It changes the microbiome of the pouch to favor firmicutes and forms a distinct niche that protects and sustains the young echidnas. This research study strongly connects to our biology class through the topics of species richness and evenness, taxonomy and lineage, and ecological niches.